Hi, everybody, and welcome to our module on nephron physiology. Shown on the screen is a drawing of the nephron. You can find millions of these in the human kidneys, and the job of the nephron is to filter the blood and make urine that will be excreted from the body, basically to take some elements out of the blood and excrete them in the form of urine and leave others behind. And the first basic principle of how the nephron works that you need to understand is that the nephron filters a lot more stuff than it wants to excrete in the urine. So blood comes into the nephron here at the glomerulus, and lots of things, water and other solutes, are filtered that the body actually wants to hold on to. And what's then going to happen is, as those substances move through the nephron, they are going to be selectively reabsorbed. So for example, things that the body wants to hang on to, like water and sodium chloride and potassium and all these things I've shown here, will be reabsorbed. And you can imagine how this evolved. Millions of years ago, our ancestors just had a filter, and lots of things that were useful to the body went out in the urine. And then over time, the nephron evolved to selectively reabsorb the useful substances, and now we're able to pick those back up and hang on to them. The way that the nephron selectively reabsorbs solutes and other substances that are useful to the body is by transporting them from the urine side of the nephron into the interstitium or blood. There are cells that line the tubules of the nephron, and they perform this job of transporting solutes out of the urine and into the interstitium or blood. The first thing you need to understand about how this works is that these cells that line the nephron are polarized. They have one side called the apical membrane. That's the side that faces the urine or the lumen of the nephron. And they have another side called the basolateral membrane. That is the side of the cell that faces the interstitium or blood. So first, let's start by talking about how a substance like sodium might be reabsorbed. And let's imagine that we have a very high concentration of sodium in the urine and a relatively low concentration of sodium inside a cell lining the tubule. If this is the case, sometimes the nephron will reabsorb a solute like this by having a protein that assists and simply grabs the sodium from the high concentration side and moves it to the low concentration side. This does not require energy because we are going from a high concentration to a low concentration. On the other hand, if we have a situation where there's a relatively lower concentration of sodium in the lumen and a relatively higher concentration inside the cell, then we can also have a transport protein that assists in this process, but that will require ATP. It will require energy because we are going against the concentration gradient. Sometimes the tubule cells in the nephron won't use any proteins at all. They'll just use simple channels. So if we have a high concentration of sodium in the urine and we have a low concentration in the interstitium of blood, then sometimes cells will just have channels or openings where sodium can diffuse in and diffuse out, and this is another way that reabsorption of solutes can occur. And then finally, in some cases, the cell won't be involved at all, but sodium and other solutes will simply be absorbed by what's known as the paracellular route. This can happen if we've got a low concentration over here and a high concentration over here. Then solutes can just pass through the spaces between cells and be reabsorbed by the kidneys. Osmotic diffusion is another method of transport used in the nephron that is unique to water. So for example, if we have solutes dissolved in the urine side of the nephron such that we create a low osmolarity of 50 milliosms, and if we have a high osmolarity of say 1200 milliosms in the interstitium or blood side, then water can be reabsorbed by osmosis. It can simply diffuse into the cell and out of the cell, or it can be reabsorbed by the paracellular route. The idea here is that osmosis is a unique process to water, and anytime we create a difference in osmolarity between the lumen and the interstitium or blood, we can then reabsorb water by osmosis. Now let's talk about the segments of the nephron. So blood enters here in this portion of my drawing called the glomerulus. And remember what I told you before, lots of things are filtered at the glomerulus that the body does not want to lose in the urine, and these things will be reabsorbed as fluid moves along the tubule of the nephron. In addition, some segments of the nephron can secrete substances into the urine. You can basically think of the nephron as 5 different segments, each doing a different job in terms of reabsorption or secretion. The first segment is the proximal tubule, then we have the descending limb of Henle, then we have the ascending limb, then we have the distal tubule, and finally the collecting duct. And in this video, we're going to go through these segments one at a time and talk about what they do. We'll start by talking about the proximal tubule. I like to think of this section as the workhorse of the nephron. It reabsorbs more solutes than any other portion of the nephron, and we'll talk about this as we go along. First of all, this section of the nephron reabsorbs 100% of glucose and amino acids under normal circumstances. These are very useful solutes to the body. Glucose obviously is an energy source, and amino acids are important for protein synthesis. The body does not want to lose these in the urine, so any that get into the nephron through the glomerulus are going to be completely reabsorbed under the proximal tubule under normal circumstances. In addition, other solutes that are helpful to the body, things like bicarb, sodium chloride, potassium phosphate, and also water, are reabsorbed in very large amounts at the proximal tubule. 2/3 of these substances are reabsorbed. So basically, right after the filter, the glomerulus, when all these things enter into the nephron fluid, immediately 2/3 of them are reabsorbed by the proximal tubule. The engine that drives the proximal tubule is a sodium-potassium ATPase pump found in the basolateral side of cells of the proximal tubule. It's very important that you understand this. This pump makes the proximal tubule work. It is always going, consuming ATP. The proximal tubule uses a lot of ATP, and this creates a low concentration of sodium inside cells in the proximal tubule. And what the cells are going to then do is use this low concentration of sodium as a driving force to reabsorb lots of other solutes and also water. As I told you before, proximal tubule cells reabsorb all of the filtered glucose under normal circumstances. Once there's a low concentration of sodium inside the cell, this is very easy to do. This creates a situation where we have a relatively high concentration of sodium in the lumen or the urine, and therefore sodium can diffuse down its concentration gradient linked to a pump which also reabsorbs glucose. And then there is another pump on the other side of the cells, and in this manner glucose is 100% reabsorbed from the urine to the interstitium or blood. All that potassium that is put inside the cells, creating a high concentration of potassium, can then leave the cells linked to chloride, and this pulls chloride out from inside the cell, and then chloride reabsorption can be linked to the excretion of anions. Anions are negatively charged substances. They have a tough time getting across the glomerular capillary membranes, and they often don't get filtered very well. So the proximal tubule cells need to secrete these substances to get them into the urine. So things like hydroxide, formate, oxalate, sulfate, these are all negatively charged molecules, and they are secreted by the proximal tubule cells linked to chloride. And the reason chloride can be reabsorbed is once again because that ATP pump is always working. And then once we get a high concentration of sodium over here from reabsorption, then we can use osmosis to reabsorb water via the paracellular route, and some of that water takes sodium chloride with it. So in this manner, we grab more water and sodium chloride from the lumen or the urine and bring it back into the interstitium or blood. Let me pause and make some important points about glucose. It's very high yield for you to understand this. As I told you before, this is completely reabsorbed by the proximal tubule under normal circumstances, and it's reabsorbed together with sodium by a co-transporter. If a patient develops a high glucose concentration in the serum, as occurs in diabetes, if the glucose level gets to about 160, then glucose will start to appear in the urine. At 350 milligrams per deciliter in the serum, all transporters are saturated, and at that point, any further increase in serum glucose leads to more glucose in the urine. If we were to plot the serum concentration of glucose on the x-axis, and if we were to plot the urinary excretion of glucose on the y-axis, what we would see is initially this would just be a flat line at 0. The reason it would be a flat line at 0 is because there's no urinary excretion of glucose until the serum glucose concentration rises to about 160. At that point, we would start to see a curve upwards as some of the proximal tubule cells become saturated and are unable to reabsorb any further glucose. Once we hit about 300 milligrams per deciliter, we would then see that this would become a straight line at a 45-degree angle. And the reason for this is because at this point, every reabsorbing transporter in the nephrons is saturated, and anytime the serum concentration goes up any further, it just increases the amount that's being excreted in the urine. And you may know that diabetes mellitus gets its name because the urine in this condition is sweet. Hundreds of years ago, people tasted it, and the reason it's sweet is because of the glucose. It is very, very high yield to know that in pregnancy, some glucose in the urine or glycosuria is normal. The reason for this is because the GFR goes up in pregnancy as the plasma volume goes up. In addition, there is evidence that there's some decrease in glucose reabsorption. And for this reason, if you want to screen mothers for diabetes occurring in pregnancy, you need to use serum glucose testing, because finding some glucose in the urine doesn't necessarily mean the pregnancy is abnormal. Let me also make a special note about amino acid reabsorption in the proximal tubule. Just like with glucose, all amino acids are reabsorbed in the proximal tubule under normal conditions. Just like with glucose, they are reabsorbed together with sodium via a co-transporter, as I've shown at the bottom of this slide. There's a clinical tie-in here with a rare disorder called Hartnup disease. Children born with this rare disorder have no tryptophan transporter in the proximal tubule. Tryptophan is an amino acid, so as a result, they lose tryptophan in the urine and they develop a tryptophan deficiency. Tryptophan is an amino acid used to synthesize the vitamin niacin, so children deficient in tryptophan from Hartnup disease will develop niacin deficiency, and niacin deficiency has a skin rash associated with it called pellagra. Pellagra consists of plaques on the skin and desquamation. So the way this will present in a board question is a baby who develops a skin rash in sun-exposed areas and then has amino acids in the urine. Remember, you should never see amino acids in the urine under normal circumstances. If you see this together with a skin rash in a baby, you should think of Hartnup disease. The problem is amino acid reabsorption in the proximal tubule of the kidneys. Now let's talk about the reabsorption of bicarbonate in the proximal tubule. There are some metabolic processes in the body that produce H plus or acid. There are other metabolic processes in the body that produce bicarbonate or HCO3 negative. However, on balance, normal metabolism produces acid every day, and we need bicarbonate around to buffer this. And as I talk about in some of the acid-base videos, our kidneys can actually synthesize bicarbonate, but before they go to all the trouble of making new bicarbonate, they want to make sure they reabsorb all the bicarbonate they can, and therefore reabsorption of bicarbonate in the proximal tubule is very important. The way this is done in the proximal tubule is through an antiporter of sodium and hydrogen ions. These hydrogen ions go out into the lumen or the urine where they combine with bicarbonate to form H2CO3. There is then an enzyme that is present in the kidneys called carbonic anhydrase, a very important enzyme, which can convert H2CO3 into carbon dioxide and water, which can then enter cells in the proximal tubule. The same enzyme, carbonic anhydrase, inside of cells can then reform H2CO3, which can then break apart into another H plus and an HCO3 negative atom. The HCO3 can then be carried out into the interstitium and blood together with sodium, and the hydrogen ion is recycled to keep this whole process going in a loop. And the net result of this whole process is that HCO3 is reabsorbed into the interstitium of blood, and it takes with it sodium, so that sodium reabsorption is linked to bicarbonate reabsorption. In addition to bicarbonate being important for acid-base balance, there are a number of other clinical correlations of bicarbonate reabsorption that I talk about in other videos. In the diuretic video, I talk about carbonic anhydrase inhibitors. These are weak diuretics because they block sodium reabsorption. They also lead to bicarbonate loss in the urine. And in the renal tubular acidosis video, I talk about a type 2 RTA, which is an ion defect where patients have the inability to absorb bicarb in the proximal tubule, and this leads to a metabolic acidosis. Fanconi syndrome is relatively rare, but it is very high yield for you to understand this syndrome for your boards because the cause of Fanconi syndrome is a loss of all of the proximal tubule functions. Basically, in this syndrome, the proximal tubule doesn't work. As a result, you see impaired resorption of solutes. Remember, as we have discussed, the proximal tubule is critical to reabsorbing many solutes that the body wants to reclaim. This includes bicarbonate, glucose, amino acids, phosphates, even some small low molecular weight proteins which can be filtered at the glomerulus. They are reabsorbed by the proximal tubule, and all of these things are going to go out in the urine in Fanconi syndrome, and they like to test you on this to see if you understand what the proximal tubule is supposed to do. The presentation of Fanconi syndrome is very similar to diabetes. That's because there is glucose in the urine, which leads to diuresis. This makes the patient urinate a lot, and then the patient becomes thirsty, so there will be polyuria and polydipsia. And in a board question, you will see a description of polyuria and polydipsia along with glucose in the urine, and you will say, aha, this is diabetes, but it's not. And the reason is because in Fanconi syndrome, there is a normal serum glucose. There is no hyperglycemia because insulin and glucagon and all the body systems to control glucose levels are working. You can also see a non-anion gap acidosis. This is because you're losing bicarb in the urine. There can be hypokalemia because anytime there is increased nephron flow, this leads to a loss of potassium, and we'll talk more about this later in this video. There can also be hypophosphatemia from loss of phosphate, and there can also be amino acids in the urine. And remember what I told you before, in Hartnup disease, amino acids should never be in the urine, and if you see a description of this, you should automatically think something is wrong with the proximal tubule. Fanconi syndrome can be inherited or acquired, and as I told you before, in general, it's rare. The inherited form, which presents in infancy, is associated with cystinosis. This is a very rare lysosomal storage disease where babies cannot break down cysteine and therefore it accumulates in their cells. If you look at the bottom of the screen, this is the amino acid cysteine. If you link two of these together, you get cysteine. And cysteine often links together to form this structure of cysteine, and you need to be able to break this down in your lysosomes. If you're missing an enzyme, you cannot break this down, and therefore cysteine accumulates all over the body. That's called cystinosis. And one of the first places it tends to accumulate is in proximal tubule cells. So for this reason, babies born with this rare lysosomal storage disease will present in infancy with Fanconi syndrome. Now, just an aside here, don't confuse cystinosis with cystinuria, even though the two names sound alike. Cystinuria is an isolated proximal tubule defect where patients have impaired reabsorption of cysteine. That's that molecule that is two linked amino acids together. The problem here is that cysteine will accumulate in the urine and a rare form of kidney stone called cysteine kidney stones will develop. And I talk about this in more detail in the video on kidney stones. Getting back to Fanconi syndrome, let's talk about the acquired causes. So lead poisoning is a classic acquired cause of Fanconi syndrome. Lead can poison proximal tubule cells. Multiple myeloma is also a classic cause of an acquired Fanconi syndrome. In the hematology section, I talk about multiple myeloma, but it often damages the kidneys and it can cause proximal tubule damage, which leads to the Fanconi syndrome. And then finally, there is a long list of drugs that are occasionally associated with Fanconi syndrome. I've listed all of them here. One of the classic drugs is cisplatin, which is used for chemotherapy. Another one is ifosfamide, which is an alkylating agent for chemotherapy. Fanconi syndrome has also been reported associated with these other drugs on the screen here. The next thing I'm going to do is go through this classic graph that you can find in renal physiology textbooks on the screen. It shows the changes in concentration of various solutes as fluid moves along the proximal tubule. So the distance along the proximal tubule is on the x-axis. On the y-axis is the concentration of solutes. However, they are expressed in a very unusual way. They are expressed in the ratio of the concentration of that solute in the tubule compared to plasma. So initially, all of the substances have the same concentration as plasma right after they are filtered at the glomerulus. But then as the tubular fluid moves along the proximal tubule, the concentration changes. Some of those solutes see their concentration rise relative to plasma, others see their concentrations fall. So we're going to try to explain this entire graph in the next few minutes here. The first thing we need to talk about is all the possibilities for how the concentration of solutes might change as fluid moves through the proximal tubule. So let's imagine we have a 50-50 mix of a solute represented by the red dot and water represented by the blue dot. And now let's imagine we move along the proximal tubule in the nephron and we reabsorb the solute, the red dots, at the same rate we reabsorb water, the blue dots. At the end of the proximal tubule, we will still have a 50-50 mix. There's less red and less blue, but the mixture is still 50-50. And this is what happens for sodium and chloride. They are reabsorbed at roughly the same rate as water, so even though we are reabsorbing sodium and chloride, the relative concentration doesn't change much through the proximal tubule. Now let's imagine we have a different solute also at 50-50, and as we move along the proximal tubule, we reabsorb lots of that solute, even more than we reabsorb water. This would leave us with a mix that is less than 50-50. In other words, the concentration of that solute would fall. And this is what happens for glucose and bicarb. These two solutes are actively reabsorbed by the proximal tubule. They are reabsorbed at a greater rate than water, and therefore their concentration falls along the length of the proximal tubule. Finally, let's imagine we have a third mix of 50-50 solutes and water, and we move along the proximal tubule. And let's imagine that none of the solute is reabsorbed at all. In other words, all the red dots remain, but we do lose water as that is reabsorbed in the proximal tubule. Well, now we'd be left with a mixture that has a higher concentration than 50-50, and this is what happens for inulin and creatinine. Inulin and creatinine are not reabsorbed, and therefore they sit in the proximal tubule and become more concentrated as water is pulled out. So now let's go back to this chart and see if we can make sense of it. So glucose, amino acids, and bicarb, they are actively reabsorbed by the proximal tubule. So their concentration falls relative to plasma as shown in this bottom line here. If the reabsorption of a solute were exactly equal to water, it would just be a flat line at 1.0. And you can see that chloride, urea, sodium, and potassium are relatively close to that line. They stay pretty close to the straight 1.0 line if there were no concentration change. Inulin and creatinine follow this straight line up here at an angle because they are not reabsorbed at all. And then finally, PAH, which is para-aminohippuric acid, actually has the steepest line of all. The reason it's like this is because it is secreted by the tubule. So not only is it not reabsorbed, but the tubular cells add PAH to the urine in the lumen, and therefore its concentration rises the greatest. Now we're going to move on from the proximal tubule and talk about the next portion of the nephron, the thin descending loop of Henle. That's this section of the nephron in my drawing here. The thing you need to understand about this portion of the nephron is that it is impermeable to sodium chloride, but it can absorb water, and therefore what it does is concentrate the urine that is found in the tubule. Basically, water leaves the urine, leaving sodium chloride behind, and that makes the urine more concentrated. And the force that draws the water out of the thin descending loop of Henle is the hypertonicity of the surrounding medullary interstitium. So let's talk about that now. Here's a cross section of a kidney, and this outer portion here is the cortex of the kidney, and that's where you find the glomeruli. However, where you find the loop of Henle is that it dives down from the glomeruli into this portion here, which is the medulla. The outer portion is called the outer medulla, the inner portion is called the inner medulla. And as the urine moves through the tubule down into that medulla, the urine becomes more concentrated, and that's because the solution bathing the nephron or surrounding the nephron in the medulla is very concentrated itself, so it draws water out of the tubule and into the nephron. This is easier to understand with a picture. So here's the glomerulus, and fluid that enters the lumen of the nephron in the glomerulus has an osmolarity of about 300 milliosms, the same as plasma. As fluid moves through the proximal tubule, its osmolarity really doesn't change very much. Even though solutes are being reabsorbed, water is being reabsorbed as well. So fluid that leaves the proximal tubule still has an osmolarity of about 300 milliosms. Then that fluid starts to move down into the thin descending limb. And as it does this, water is being pulled out. And the reason water is being pulled out is because the osmolarity in the outer medulla and the inner medulla is very high. The osmolarity goes up to about 600 in the outer medulla and as high as 1200 in the inner medulla. This is a powerful force for drawing water out by osmosis. And what's going to happen is the fluid in this lumen, when it gets all the way down to the bottom, is also going to have an osmolarity of 1200. The tubular fluid is very concentrated because it's now in equilibrium with the concentrated fluid surrounding the loop of Henle in the inner medulla. It's very high yield for you to understand which solutes are responsible for maintaining that high osmolarity as high as 1200 in the medulla of the kidneys. The three solutes that are responsible for that osmolarity are sodium chloride and also urea. Urea has a very important role to play in the kidneys in terms of maintaining a high osmolarity in the medulla. Urea is generated in the liver by protein metabolism, and I talk about the urea cycle in the biochemistry section. It then travels from the liver to the kidneys where it's reabsorbed by the collecting duct. The collecting duct has a very high permeability to urea, and I'll talk more about how this works later. And then that urea settles into the medulla in the interstitium, and that's essential for maintaining those osmotic gradients. So I'll go through this once again just to make this very clear. Fluid leaving the proximal tubule has an osmolarity of about 300. It's in equilibrium with the cortex of the kidneys. Then as fluid moves down into the loop of Henle, water is removed, drawn out by those high osmolar gradients made by sodium chloride and urea, so that the tubular fluid reaches an osmolarity of 600 when it's in the outer medulla, and then when it gets down to the inner medulla, that fluid is very concentrated as high as 1200. Once urine reaches the bottom of the loop of Henle and has become very concentrated, it then begins to move up the ascending limb of the loop of Henle. And I told you before that the descending limb is permeable to water and impermeable to sodium chloride. The ascending limb is the opposite. It is impermeable to water, however, it is permeable to sodium chloride. What this means is as the urine moves up this ascending limb, sodium chloride is going to be removed. This is going to pull solutes out of the tubular fluid and dilute it all the way down to a very low osmolarity of 120 milliosms at the top of the ascending limb of the loop of Henle. You'll also notice in this drawing that the ascending limb has two portions, a thin portion and a thick portion. The thick portion is the most important portion. That's where sodium chloride is actively resorbed to take that tubular fluid all the way down to a very dilute and low osmolarity of 120. Sodium and chloride are reabsorbed in the thick ascending limb of the loop of Henle by a very important pump that reabsorbs sodium, potassium, and two chloride ions. This is the same pump that is inhibited by loop diuretics, which I talk about in the diuretic module. The reabsorption of sodium is an energy-dependent process. There is a sodium-potassium ATPase pump found in the thick ascending limb. It reabsorbs sodium and creates a low concentration of sodium inside the cell that can be used to drive the activity of this pump right here. Now, some of this potassium that's reabsorbed by this pump will leak back out into the tubular fluid. This creates a positive charge in the urine in the lumen of the nephron at this portion of the loop of Henle. That positive charge is very important because it is used to drive the reabsorption of other positively charged ions like magnesium and calcium. They are reabsorbed via the pericellular route, and they are driven out by the positive charge in the lumen of the nephron. Moving on, now we'll talk about the distal tubule of the nephron. That's this section shown in my drawing down here. It's the section of the nephron that comes right before the collecting duct, which is the last portion of the nephron. This part of the nephron resorbs sodium and chloride. Once again, there is an ATP pump that reabsorbs sodium in exchange for potassium. This creates a low concentration of sodium inside the cell, and that low concentration of sodium is used to reabsorb sodium and chloride. The chloride can then diffuse out a pump on the other side of the cell. The distal tubule is also very important for calcium reabsorption. Calcium can enter distal tubule cells via a channel and then be reabsorbed in exchange for sodium. And this is important because, as I'll talk about in other videos, parathyroid hormone acts on the distal tubule to drive more calcium reabsorption through these channels and transporters. Finally, we get to the collecting duct, the last portion of the nephron. There are two cell types found in the collecting duct. The first cell type is called a principal cell, and the second cell type is called an intercalated cell. The principal cells have an ATP pump that generates a low concentration of sodium inside the cell. In this manner, sodium can be reabsorbed by the principal cells via a channel. This channel is called an ENaC channel. ENaC stands for epithelial sodium channel, and we'll talk about later how this is an important channel because its activity is regulated by aldosterone. In addition, potassium enters the cell via the sodium pump and then can exit into the lumen via a potassium channel. And so in this manner, the collecting duct is important for the secretion of potassium in the setting of high potassium states in the body. And then finally, the principal cells are also important for the reabsorption of water. And I'll talk more about that in a minute because this reabsorption of water is modulated by antidiuretic hormone. The main role of the intercalated cells is to secrete acid. They secrete hydrogen ions into the urine via an ATP pump. So here I've summarized some key points about the collecting duct in terms of its functions. It reabsorbs sodium and water, and it secretes potassium and hydrogen. And the one thing you need to understand is that anytime there's increased sodium delivery to the collecting duct, there will be more potassium excretion. That's because potassium goes out in the collecting duct in exchange for sodium coming in. And more sodium gets reabsorbed when you send more sodium to the collecting duct. So for this reason, hypokalemia often occurs in patients on diuretics like loop diuretics and thiazide diuretics. That's because these drugs send more sodium to the collecting duct, which then gets exchanged for potassium. I'll go back to this slide to make this point clear. So if you send more sodium to the collecting duct, more sodium will enter through this channel and go out through this pump. Then more potassium will come in and go out through this channel. So the two are interrelated. More sodium going in this direction always turns into more potassium going out in that direction, and that's why any drug that promotes diuresis and sends more sodium to the collecting duct can potentially cause hypokalemia. A special feature of the collecting duct is that its activity is heavily influenced by two hormones, aldosterone and antidiuretic hormone. Both of these hormones act on the kidneys and they regulate the function of the collecting duct. Aldosterone is a steroid hormone. It has a structure that is derived from cholesterol, just like all steroid hormones, and this allows it to pass through the lipid bilayer of cells and have effects inside of cells, including on gene expression. It's called a mineralocorticoid because it has effects on minerals like sodium and potassium, and this is in contrast to cortisol, which is a glucocorticoid and has effects on blood sugar. Both cortisol and aldosterone are released from the adrenal cortex, and I talk about this in more detail in the videos on the adrenal gland in the endocrine section. Aldosterone's effects on the collecting duct are summarized on this slide. It can increase the number of sodium-potassium ATPase proteins. It can increase the number of ENaC sodium channels in principal cells. It can also promote potassium secretion by principal cells and hydrogen ion secretion by intercalated cells. Here's a slide showing the effects of aldosterone on the principal cells. The number of sodium-potassium ATPases can be increased by aldosterone. Remember, it's a steroid, so it can travel inside the cell and induce gene expression, so it can increase the expression of genes that code for proteins like the sodium-potassium ATPase. It can also increase the number and activity of the ENaC sodium channels, and it can also increase the secretion of acid or hydrogen ions from intercalated cells. The overall effect of aldosterone on the collecting duct is that it will increase sodium and water resorption. This will increase the effective circulating volume, and that's why aldosterone is part of the renin-angiotensin-aldosterone system. The job of that entire system is to respond to low perfusion pressure in the body and increase the circulating volume. Aldosterone will also increase potassium excretion, and that's why it is a hormone responsible for the regulation of serum potassium levels, and it will also promote acid excretion. Its release is stimulated mainly by angiotensin II and by high potassium. So angiotensin II is how the renin-angiotensin-aldosterone system stimulates aldosterone release. And then a second trigger for its release is a high potassium level in the serum. If you consume a high potassium diet, your body will begin releasing more aldosterone to bring the potassium level back down to normal. Finally, ACTH, which acts on the adrenal gland to promote cortisol release, can promote some aldosterone release, although this is a minor effect. Now we're going to talk about antidiuretic hormone, or ADH, and in order to understand how this hormone works, you need to understand the different sections of the nephron and their permeability to water. So the proximal tubule and the descending limb of Henle are permeable to water. Lots of water is reabsorbed in these segments, as we discussed earlier in this video. The ascending limb and the thick ascending limb and the distal tubule are impermeable to water. No water is reabsorbed in these portions of the nephron. And then the last section of the nephron is the collecting duct, shown here. The water permeability of this section is variable, and it all depends on antidiuretic hormone. Antidiuretic hormone is often called ADH. It is also sometimes called vasopressin because it can raise the blood pressure. It's a large peptide hormone. Note that it has a very different structure from the steroid hormone aldosterone. And what ADH does is it promotes free water retention and inhibits secretion of free water by the kidneys. There are two receptors for ADH in the body called V1 and V2. V1 is the receptor found peripherally that is responsible for ADH's vasoconstrictive effect that can raise blood pressure. The V2 receptors are found in the collecting duct, and they are responsible for the antidiuretic response of ADH, which we'll talk about in a minute. This hormone is released by the posterior pituitary gland, and there are two stimuli for release of ADH. You need to understand that ADH increases the amount of water in the body, so you want it to be released anytime the body needs more water. The major physiologic stimuli under normal circumstances is hyperosmolarity. So anytime the osmolarity of the serum goes up, ADH is released to dilute the osmolarity back down to normal. And on a normal day-to-day basis, this is the major stimuli for release of ADH. There is, however, a second stimuli that is important in many disease states, and that is volume loss. This is called the non-osmotic release of ADH because it does not depend on serum osmolarity. Basically, if your effective circulating volume gets low enough, that decrease in ECV will be detected by the pituitary gland, and it will release ADH no matter what the serum osmolarity is. This is a life-saving intervention. Basically, the body is detecting dangerously low circulating volume, and it's ignoring the serum osmolarity and just releasing ADH to try and conserve and hold on to water. The way that ADH stimulates water resorption is by acting on V2 receptors in the principal cells of the collecting ducts. These receptors are G-protein linked with a cyclic AMP second messenger system, and once the V2 receptors are activated by ADH, an endosome is inserted into the cell membrane. I'll show you a picture of that in a second. The endosomes contain a water channel called aquaporin 2, and as a result of these channels being inserted into the principal cells, there's more permeability of those cells to water. So here's a picture of how this works. So ADH comes along and binds to a V2 receptor. That leads to the insertion of channels on the luminal side of principal cells called aquaporin 2 channels. Once more of these channels are in place, more water enters the cell, and then there are other aquaporin channels like 3 and 4 that are always active on the other side of the cell. So basically, the amount of water being resorbed is controlled by the number of these aquaporin 2 channels, and the level and function of those aquaporin 2 channels is under the control of ADH. So the collecting duct can basically be completely permeable to water or completely impermeable to water. It all depends on the effects of ADH. So let's imagine that you are water deprived. For example, imagine you've been walking outside on a hot day, you haven't had any water to drink, and you've been sweating profusely. In this setting, your ADH level will get very high. That's because your plasma osmolarity is rising and it's being sensed by the pituitary gland, and ADH is being released. This is going to make the collecting duct here permeable to water through insertion of aquaporin channels. As a result, as tubular fluid moves down, it will equilibrate with the surrounding interstitial osmolarity. So that fluid osmolarity will rise to 600 in the outer medulla and to 1200 in the inner medulla. As a result, you will excrete very concentrated urine with an osmolarity of 1200 milliosms, and that's appropriate. You don't want to secrete much free water. You want to secrete mostly just solutes and hang on to all the free water you can. And that's what happens in water deprivation when ADH levels are high. In contrast, if you consume a high water intake, for example, if you chug 8 glasses of water, your ADH level will get very low. This will make the collecting duct impermeable to water. Now, as that tubular fluid moves down the collecting duct, some sodium chloride resorption continues to occur, driven by those pumps we talked about before in the principal cells. As a result, you will make urine that has a very low osmolarity, like 60. In other words, you are excreting mostly free water and very little solutes, and this is what you want to do. There's excess free water in the body, so you want to excrete urine that is very dilute and has a very low osmolarity. Earlier in this video, I told you that urea is a very important osmol for maintaining those osmotic gradients in the kidney along with sodium and chloride. I also mentioned earlier that the collecting duct is permeable to urea, and that's how urea gets into the interstitium. In particular, the collecting duct in the medulla, the medullary collecting duct, is permeable to urea. You should also be aware that urea reabsorption is linked to ADH. ADH increases the amount of urea resorption by the collecting duct and allows urea to enter the medullary interstitium to maintain those osmotic gradients, and then the urea is recycled by going back into the descending limb via transporters. This is easier to understand with a picture. So the first portion of the collecting duct shown here is permeable to water, but not to urea. This draws water out in the setting of high ADH levels. As a result, when you get down to this bottom half of the collecting duct, there is a very high concentration of urea found in the tubular fluid. That urea will then be absorbed along with water, and that adds urea to the interstitium. Transporters in the thin descending loop of Henle will pick the urea back up, put it back into the fluid where it'll go back around and then be recycled. This is called urea recycling in the kidney. But the key point here to remember is that urea is reabsorbed in the collecting duct, that it is influenced by ADH, and that urea is a very important osmol for maintaining those high osmotic gradients that you need out here in the medullary interstitium so that water can be reabsorbed. So here's a summary of the major functions of the collecting duct. It reabsorbs sodium and water, and this depends on ADH for water reabsorption, and it's under the influence of aldosterone for sodium reabsorption. The collecting duct also secretes potassium and acid, and this depends on aldosterone, and the collecting duct is also important for urea resorption. I'll finish this video with a couple of summary slides showing you where water and important solutes are reabsorbed in terms of portions of the nephron. So 2/3 of sodium is reabsorbed by the proximal tubule. Remember that the proximal tubule is the workhorse of the nephron. It reabsorbs most of all the solutes that are important in the body. You then reabsorb about 1/4 of the sodium in the thick ascending limb through those transporters we talked about, and the rest is reabsorbed in other portions of the nephron. So remember, for all the discussion that goes into aldosterone and ADH and all the reabsorption of sodium in the collecting duct, that's only a tiny portion of your sodium. Most of it is reabsorbed earlier, especially in the proximal tubule. And just like sodium, most of the water that the kidney reabsorbs is reabsorbed in the proximal tubule, about 15% in the thin descending limb, and the remainder can be reabsorbed in the collecting duct, but this all depends on whether the body wants to hold on or excrete free water, depending on the level of ADH. And that concludes our video on nephron physiology.